Welcome, everyone. I'm Mark Johnson, a volunteer with the Canadian Celiac Association. I'm going to be your host today. I'm pleased to be joined by Dr. Elena Verdu, a professor in the Division of Gastroenterology at McMaster University. Welcome, Dr. Verdu.
Thank you, Mark.
So this is the second in our In Conversation series for you on matters relating to celiac disease and the gluten-free diet. Every other month, we're going to be bringing these to you as part of our CCA Connects newsletter. We're talking to leaders and change makers in the gluten-free industry, research field, medical professionals, stakeholders, and more. We hope that you will continue to listen and learn, and you can always share your thoughts on potential guests with us too. Just send an email to communications@celiac.ca with any ideas. So we're very excited to have Dr. Verdu with us today. She's a medical doctor and a professor at McMaster University. Dr. Verdu trained in internal medicine at the University of Buenos Aires and as a clinical research fellow in gastroenterology at the University Hospital of Lausanne in Switzerland. She earned her doctorate in immunology at the Czech Academy of Sciences in Prague. Dr. Verdu is also a past member of the CCA's Professional Advisory Council, and she has been the recipient of funding from the J.A. Campbell Research Fund of the CCA. Her research interests focus on host microbial and dietary interactions in inflammatory gastrointestinal disorders such as celiac disease and IBD. She holds the Canada Research Chair in intestinal inflammation, microbiota, and nutrition. I'm thrilled to be able to speak with her today. So let's get started. Dr. Verdu, could you first tell us a bit about the research that you're doing at McMaster University, especially your work on gut microbiota and gut health?
So thank you very much, Mark, for that introduction. And I'm also very excited to be here. So look, we need to understand that there are two necessary factors for the development of celiac disease, and this is gluten and genes. But although they are necessary, they're insufficient. And so my research interest has to do with that, with trying to identify and decipher the additional environmental factors that could play a role in increasing the risk to develop celiac disease in a person that has the right genes. And we think that those factors are microbial in origin. And so what we have found is that patients with active celiac disease have certain bacteria in the duodenum that can metabolize gluten. And they metabolize it in a different way than bacteria that we mainly obtain from individuals that do not have celiac disease. And so this metabolism of gluten leads to more peptides with more immunogenicity. That means that stimulate more immune responses. And other bacteria that we can obtain, for example, from the healthy duodenum, can completely detoxify those peptides. So it's the community of bugs that inhabit the intestine that can lead to this different pool of peptides with more or with less antigenicity. And so this is the core of my work in the lab with respect to celiac disease.
So to break it down a bit, essentially, you said the bacteria that's in the duodenum, which is part of our intestinal tract, part of our digestive system, you're thinking that it might be acting a little bit differently in people with celiac disease compared to others?
Right. And it is not only about metabolism of the main trigger, which is gluten. For example, the bacterial metabolism of patients with active celiac disease sometimes is directed towards other dietary sources. So for example, an amino acid that is called tryptophan. This amino acid is very important. It's an essential amino acid we obtain with the diet. And when our gut bacteria metabolizes this substrate, it produces certain molecules, they are called indoles, that are immunoregulatory. And what we have found is that some patients with active celiac disease have a microbiota that cannot produce these immunoregulatory metabolites from tryptophan. So it's exactly as you say, the functionality of the microbes, that it's not always what microbe you have, but what they are doing in the celiac duodenum may be different and may be contributed to a pro-inflammatory environment.
When you say active celiac disease, do you mean for those who have been untreated?
Exactly, those untreated, those that are consuming gluten, but your question is very relevant because a proportion of patients that go on to develop that are diagnosed with celiac disease and go on to follow a gluten-free diet will not completely heal. And so we could be referring to those subjects, to those patients that although they are on a gluten-free diet, they still have some active disease.
Mm-hmm. Okay, sounds like another reason for people to strictly follow the diet. So looking forward, where do you see this research is going? Like, what's the end point of these studies?
So the reason why we do this is because we need to understand mechanisms that can be then used to prevent a disease or to better treat it. So this example that I was giving you of patients that go on to develop, that are diagnosed with celiac disease, they perform a gluten-free diet, but still they are symptomatic, or their villus to crypt abnormalities are not completely normal, even if they are on a strict gluten-free diet, because it's very difficult to be completely gluten-free. And so we can target this population if we know the mechanisms that are driving that low-grade inflammation in these patients.
Could this be linked to, like, some people with celiac disease say, well, I still have problems with oats, or I can't digest corn properly, lactose, of course. Like, could your research be applicable to this kind of problem?
Yes, you are very right. And actually, we have done some work on other immunogenic proteins in wheat. So, for example, amylase trypsin inhibitors. These are non-gluten proteins, but they are contained in different cereals. And so ATIs, what we call them, amylase trypsin inhibitors, could also stimulate some immune responses. And they are also very poorly digested in our gut. And so these concepts of lack of proper metabolism of dietary substrates could apply to other triggers.
And I know you're a world-renowned researcher in the celiac disease world. More broadly, if you're able to, what do you hope to see in celiac disease research worldwide in the next 3 to 5 years? Like, where's the greatest potential?
So, one thing that I think where the area might be going in the future relates to understanding the different phenotypes, clinical phenotypes in celiac disease. So why some individuals that are genetically predisposed are never going to develop the disease and others will. In fact, it's the majority that don't. So why patients that already have high antibody titers against TG2 will never develop it, and this is what we call, potential celiacs, and others will develop it. So all these differences in phenotypes, in clinical presentation, are very interesting to study, because we might be missing here the additional factors that are making that patient go on to have full-blown celiac disease or stay without tissue destruction. So I think a lot of the efforts are going to go into understanding what causes these different disease phenotypes. And we can do this with long longitudinal studies, and these are very important studies, but they are difficult to do, they are expensive, we have to wait many, many years. And the other one is, where I am involved with, which is a translational part, where we take these samples from these clinical cohorts and we try to model things in the lab. And so a lot of modeling, I think there's going to be a lot of effort into doing this in the lab with animal models, with what we call the mini guts, which are mini guts that we can obtain from stem cells, from patients, from animals, and trying to answer specific questions related to why this happens.
So if I understand correctly, so we know that certain segments of the population have the HLA, DQ2, and DQ8 genes. So if anyone out there has had one of the gene tests, that's the genes that they're looking for to see if you have the potential to develop celiac disease. But what you're saying, Dr. Verdu, is only a small number of people with those genes actually develop the disease. And you're trying to understand, the research world is trying to understand why certain people go on to develop it while most don't. There might be other factors at play.
Exactly. And even those that have potential celiac disease, which already have high antibodies. So this is a very interesting population where they don't have active celiac disease, they don't have the damage in the intestine, but they do have this adaptive immune response. They lost already the tolerance, despite the fact that they are healthy from the gut point of view. So this is, it's very interesting to understand why some of these will go on to do the intestinal damage and others won't. And if I may say, the other important area of future research is, of course, therapies, right? Therapies that we can add to the gluten-free diet. So there's a lot of pharma investment into this area.
I'm sure some of the new parents out there are thinking, oh, well, my children have been tested, they have the genes. Is there anything I can do to prevent them from getting celiac disease? Are we too early in the process, or is there anything that you can suggest?
Yes, it is too early. It is too early to say this is what you should do, this is the probiotic you should take. We can't give those recommendations right now. What the patients need, in this case, need to do is to know that their children are genetically susceptible, monitor that, and have the kids follow up by a health professional.
And there are studies going on with the models that you mentioned, looking at those who do develop celiac disease and those who don't, and what are the factors. So like this stuff can be pretty complicated for the layman out there. Can you tell me, like, why is the research that is going on at your lab, for example, important for people with celiac disease? Like, how does it impact them?
Well, you know, celiac disease is a very misunderstood disorder, not only, you know, among the general population, but at the level of the government, and even sometimes at the level of health professionals that are not specialized in celiac disease. So the research that we do puts celiac disease on the map. And this is very important because we have 300,000 at least Canadians that are suffering from celiac disease. This is not a benign disease as generally considered. And so we need this research to explain and inform our colleagues that celiac disease is an autoimmune disorder that needs to be supported at all levels, institutional, governmental, and at the patient level. And I don't know if you would like me to elaborate here more, but this, for example, I can give some examples. There is lack at the institutional level of centers that are specialized for the care and follow-up of celiac disease. And our center for adult patients in McMaster is the only one across Canada. This should not be like this. And at the government level, you have the example of Ontario being a large province that is not covering the TG2 test outside of the hospital system. So this illustrates how important it is that we have research in celiac disease to answer these misunderstandings and to educate at the level of the institution, healthcare professionals, public health, government, that this is a disease that we need to care about.
You mentioned the celiac clinic at McMaster. Could you quickly elaborate on what that involves for anyone out there who might not be aware?
Right. So this is a celiac clinic that is directed by Dr. Inés Pinto at McMaster University, and of course it has the primary objective of caring for the adult population of patients with celiac disease, because we realized many years ago that this didn't exist. Most of the clinics are pediatric, and when that patient crosses the magical line of 18 years of age, they are lost to our system. And as you well know, the problems don't stop there at 18 years of age or even after diagnosis of celiac disease. So this is the primary aim of the clinic. But of course, it has a research component, and that's where I'm involved with, and that translational research, taking those observations of the clinic into the basic research lab, doing the modeling to try to understand mechanisms, and then going back to the clinical setting. And the other important component of the celiac clinic is that we can get involved into the testing of therapies that are being developed by pharmaceutical companies, for example. But from my perspective, my point of view, I'm more interested in those discoveries that we do in the lab and that then we could implement, independently of pharmaceutical development. This is research-oriented research that we can implement as a discovery therapy in small populations of patients. And this is not always easy to do.
Mm-hmm. Yeah, so for everyone watching out there, so it's a clinic at McMaster University in Hamilton where that's specifically there for the treatment of patients with celiac disease. So I know a lot of our members have gone there, and it might not have been as well known that that the data gathered there are helping to support Dr. Verdu's research into celiac disease to ultimately contribute to hopefully to therapies down the road. So another reason to support that clinic. At McMaster, are you able to, like, I know you have research assistants working with you, a whole team. Can you tell us anything about any up and coming young Canadian researchers in this area?
Yeah, so you have, I already mentioned Dr. Inés Pinto. So she's a clinician researcher. Then there's who is directing the Celiac clinic. Then there is Alberto Caminero, who was a postdoc in my lab. And now he's an independent researcher and leading his lab. So Alberto has worked in many of these studies that we have been discussing before. And then there is Dr. Heather Galipo. She is a research associate in my lab. And she's at that difficult moment, trying to make the switch between being a research associate and moving into an independent career in the future. And Heather is a basic scientist, so she complements the group as the basic modeling person in celiac disease.
Is there any other research going on at McMaster University in celiac disease, or is it mostly in your clinic?
So we are. Oh, you mean in general in the university? We are. Yeah, I think we are the main group driving the research in celiac disease.
And have you been able to continue, like during COVID-19, people working remotely if possible? Have you been able to found ways to continue the research, or has it been in person with precautions? How has that worked?
So the research has continued, but we are following the mandate from the university in that sense. So it has continued as a reduced pace, where we have our shifts where we can't surpass a certain amount of trainees in the lab at a given time, but the projects have continued. The clinical research has had some strong hit, of course. We cannot just do clinical research for the moment. This we are hoping is going to resume when we are all vaccinated. But it hasn't been a complete stop of activities. We have continued to do research and to publish and to continue to do things.
I'm glad to hear that you've adapted. On the matter of research funding, I know the CCA has awarded you, along with other McMaster researchers and students, funding from the J. Alexander Campbell Research Fund of the CCA. Can you speak a bit about how these grants contributed to your work and to the advancement of our knowledge of celiac disease?
Yeah, so look, the J.C. Campbell Award was the first independent award that I received as a young investigator. So it really set me up to start with the animal models that we've been talking about. It was very difficult for me to convince, for example, the federal funding agencies that celiac disease was an important disease to study. And it was completely different how my project was taken when I submitted it to the CCA. It was immediately understood. And if it wouldn't have been, I always consider the JCA Campbell Award that it started like a snowball effect, because it allowed me to have preliminary results, to demonstrate and to ask for federal funding and to solidify my base, It also provided my first students, because I had 2 or 3 students that were funded with the smaller awards. And many of those students came through the CCA. They were members of the CCA. So it hasn't been only the funds, that I obtained initially, but also the connections that I got with students, with Decker Butzner, with Shelley Case. Those two were always introducing me to the big shots in celiac disease in the first celiac meetings I attended. So it has been more of a mentoring. I feel it like a mentoring role of the CCA for me until my research took off.
And so it shows, like, when people make a donation to the CCA, these are the kind of results that you see that it flows through and produces research that's helping to identify, like, why are some people getting celiac disease and others are not. Very, very important. I remember when you presented at one of our conferences a few years ago, and about presenting the mouse model that you're using. Is that the research that came out of initial support of the CCA?
Exactly. The initial support was to establish the mouse model in my lab, and that has allowed everything else down the road. And if I may add, also think about the students that then got support from the CCA. So many of them today are gastroenterologists, pediatricians. I have some of those that have went on to do medical school and are still being trained. And those are young physicians that are going to care for celiac patients. So, it goes beyond just supporting a research project.
Yeah, definitely many spin-off benefits. One item I didn't jump into yet is you mentioned the animal models, the mouse model. That most of us love animals, and that's like, what's going on with that? So as I recall, you're taking some of the bacteria that we discussed that are present in people that could develop celiac disease or have active celiac disease, taking some of the bacteria and putting it into the digestive system of the animals to then use that as a model for what happens. Could you explain a bit more about the logistics of how that works?
So these are mice. These are not any kind of mice. These are mice that express the celiac risk genes, right? Because we know that is absolutely necessary to develop celiac disease. So if we don't have those genes in the mouse model, the mouse doesn't react to gluten. And so it's a very special model. In addition to that, what we have done is to remove all the bacteria from the gut. And this is something that we call derivation into germ-free conditions. And we have a big facility in McMaster, the Axenic Autobiotic facility, that is dedicated to breeding mice without any kind of bacteria. And so what we can do there, they're sterile from the point of view of microbes. What we can do is then humanize these mice, which already have the human celiac risk genes, but humanize them now from the microbial point of view. So they don't have mouse bacteria, they have the bacteria that we obtain from the celiac patients or from healthy populations. And then we can introduce gluten to that. And now we have a system where we can ask questions on how these bacteria metabolizes gluten, what happens to those metabolites in terms of how they stimulate immune responses and so on.
And the what makes you choose mice? Is the digestive system of the mouse similar enough to that of humans that it's worth studying? Like what led to that decision?
It's a model, and mice are mice, and men are men, and humans are humans, right? So of course there are differences, but these are the more practical models that we can have to study diseases. And so, for example, if I make the parallel to inflammatory bowel disease, there are a variety of mouse models where we can study, pathogenic mechanisms of disease related to IBD. And so this has been lacking in celiac disease for many, many years. And so we are now at a stage when we are starting to develop disease-relevant animal models based on the transgenic expression of these genes. You know, clinical studies are super important, and they are the how can I say, the last word in research. But there are things we cannot do in humans. Sometimes these patients already have developed the disease, and to understand whether a factor is cause or consequence of the disease is very difficult in the clinical scenario. And this is where animal models come to help. We cannot just do research in animal models. We have to have a variety of models, and we have to constantly go to and fro from the basic to the clinical situation. This is what is called translational research.
And the data that you gather at the clinic from people, does that help to confirm some of the findings in the lab and vice versa?
Exactly. And we are constantly doing this movement from the basic science to the clinical scenario and back.
Okay. Oh, that's very interesting. And it must have to be a super sterile, protected environment if you're trying to not introduce any other bacteria at all into the environment.
Oh yes, this is a facility that is all biobubbled, all sterile. There is positive pressure in each room, and the mice are kept in bubbles themselves. So I don't know if you're aware with a Bubble Boy story, but this is how the technology arose. It was created for that boy that was born with a very deficient immune system, and that a simple bacteria could kill him. And so he lived many years in that bubble, and that was then translated to what is called gnotobiotic technology for mouse modeling.
Interesting. Yeah, multiple applications there. At least the mice shouldn't have gotten COVID-19 then. They're okay.
No. And it's the best place to pass the pandemic, to go to that unit.
Well, I'm sure they thank you from the bottom of their hearts. I understand, just to change gears a little bit, you recently participated in events for Digestive Diseases Week. Is there any recent research that came out of there that you could talk about with us?
Oh, so you're speaking about the Canadian Digestive Disease Week, right? So it was the 25th anniversary of the Canadian Association of Gastroenterology, and we have the online meeting, and we had one session dedicated to celiac disease, and it was chaired by Dr. Caminero and by Dr. Inés Pinto. So there were two. It was a very interesting session. I think it was attended by 300 people virtually. The topics were basically on therapies for celiac disease. So Dr. Karen Kelly and Dr. Daniel Leffler were main speakers of the session. And Daniel Leffler from Takeda was presenting his work on the new vaccine proposal for celiac disease, which is based on nanoparticles, so encapsulating gluten in nanoparticles so that they would stimulate the proper immune response to gluten in patients with celiac disease. So so it's still not something that is out there for the treatment, but it's still in development. And so we were very excited to hear about the results of those studies. And then Dr. Caminero presented work from our lab, so related to microbes in celiac disease. And then there was a medical student from Dr. Pinto, who presented work from our recent work with COVID and celiac disease, where they showed in an international survey study that we can reassure patients with celiac disease that they are not more susceptible than the general population to contract COVID.
Yes, we were very interested to see that research. A lot of questions came in. And vaccines certainly top of mind for everyone. So great to hear that research continues on potential vaccines for celiac disease. And I know that the International Celiac Disease Symposium is coming up. It was planned for Italy. I'm not sure if it's going to be in person or virtual, depending on COVID. I've attended a couple of these myself in the past. I think I heard you speak at one, actually. You learned so much there. Is there anything exciting that you're hoping to see at ICDS this coming October?
Oh, these meetings are always exciting, Mark. We take 2 years to build up the research and the clinical studies, to be presented at that meeting. So I know this has been a slow year with COVID, but I'm expecting very, very exciting presentations, especially related to results of perhaps of new clinical studies, but also of development of models. This is the area where I'm interested in. So maybe those mini guts I was telling you about. I highly recommend these congresses because they are both geared towards the researchers, but also to the patients. And it's this interaction that I think it's fantastic because we need to hear from the patients as well.
Mm-hmm. Yeah, the CCA tries to attend as well. There might be a couple from the professional advisory council, so that we try to bring some of that information home to those who subscribe to our newsletter. Okay, did you have anything else you wanted to add today?
Maybe I want to appeal to all the members of the CCA to get involved. We really need you. We need you to get involved, as I was saying, not only at the community, but at the government and institution level. We need your help. We need your help to make all these levels, understand how important celiac disease is, that we need to fundraise, we need to make noise about it, and really we can't give up. We can't give up.
I think this conversation has reinforced for all of us the importance of research on celiac disease. It helps all of us in the end. Like when these contributions come in, it's helping create a better future for everyone with celiac disease. And if anyone out there, if you're looking to make a contribution to support research on celiac disease, you can make a donation on the CCA's J.A. Campbell Research Fund. Just go to celiac.ca, click on donate and select research. You'll be helping to support projects like Dr. Verdu. Well, thanks very much, Dr. Verdu, for taking the time to join us today. Really appreciate it.
Thank you, Mark.
Okay.
Stay safe.
Thank you. You too. So I hope you've enjoyed this in-conversation session. On the matter of research, the CCA Board of Directors recently made a $30,000 contribution to the J.A. Campbell Research Fund to support Canadian celiac disease research. And we're going to be announcing this year's recipients of our awards from that fund on May 1st to help kick off Celiac Disease Awareness Month. So thanks again for joining us today. Our next event in May is going to feature Janet Bolton, President of the Canadian Celiac Association. Please watch your CCA Connects newsletter and the website celiac.ca for updates on this and other news and events. Goodbye, everyone.